be talking about the effects of molasses on a lactic acid bacteria stream known as Lactobacillus acidophilus. Before I begin, I would like to introduce myself. My name is Payne. I'm a student at George Washington High School, and I plan on attending the University of Virginia and majoring in human biology. I chose this topic because it can have important benefits for lactose-intolerant individuals, and a few of my friends are. Lactic acid bacteria also plays an important role within the dairy industry because it is responsible for the taste and smell of many products such as cheese and yogurt. Lactic acid bacteria has also been shown to be useful for recycling because it is used to create polylactic acid, which is then further used as an alternative to plastic. In addition to these, lactic acid bacteria can also be useful in fertilizer as it helps aid in plant growth. Lactic acid bacteria has multiple effects on industrial application. It is proven to be successful in helping lactose-intolerant individuals to digest lactose. There is also evidence that antibiotics have been overused when supplementing animals, which is causing antibiotic resistance when humans digest these animals. However, studies have shown that probiotics can be substituted for antibiotics and serve the same purposes. Lactic acid bacteria faces the limitation of being costly to grow due to all its specific nutritional requirements. That is why the main goal of the field is currently to bridge the gap between laboratory growth and production within the industry. It is expensive to produce lactic acid bacteria on a large scale due to these specific nutritional requirements. This is known as fastidiousness, which refers to how specific and accurate the growing conditions must be in order for lactic acid bacteria to grow. Lactobacillus acidophilus is an example of this. That is why it is imperative to ensure that the components found in MRS agar meet the nutritional requirements of the bacteria strain being grown. In addition to this, it is also important to make sure that temperature and pH level are monitored to ensure stable growth. Due to high costs and a lack of transfer between the laboratory and the industry, studies are exploring the use of alternative growth substrates, such as molasses. Molasses is a viable substrate because of the high amounts of sucrose that are found within it, which is why I chose to use it. It is a more affordable alternative to the traditional ingredients found in MRS agar. Along with lactic acid bacteria needing a carbon source, there are also amino acids that are essential to growth. Without these certain amino acids that are found within MRS agar, growth will cease. Incorporating molasses and glutathione into lactic acid bacteria growth may greatly reduce costs in the future. This is possible if the number of colonies grown is still large with inexpensive supplements. This study seeks to test the effects of molasses supplemented with glutathione on the bacterial growth of Lactobacillus acidophilus to test if molasses aids in bacterial growth. If the results show that molasses promotes more growth as opposed to only glutathione, then this study can be implemented in the dairy industry. Affordable growth substrates will benefit plants, humans, and animals on a daily basis if production is more efficient and affordable. This leads me to my research question, which is, what is the effect of amino acids supplemented with molasses on the bacterial growth of Lactobacillus acidophilus? My null hypothesis is, if Lactobacillus acidophilus is supplemented with molasses and amino acids, then there will be no difference in bacterial growth. My alternative hypothesis is, if Lactobacillus acidophilus is supplemented with molasses and amino acids, then there will be a difference in bacterial growth. I chose to formulate my hypotheses this way because after researching growth substrates and the role they play on bacterial growth, I believe that molasses would promote more growth. For my experiment, my independent variable was whether or not MRS agar was supplemented with molasses. This was so that I could monitor the growth of Lactobacillus acidophilus to see if molasses promotes more growth as opposed to only glutathione. My control group were the dishes that received MRS agar and glutathione, and some constants were the agar type used, the length and temperature of incubation, and the type of petri dishes that were used. The first thing I did was gather all my necessary materials, which consisted of Lactobacillus acidophilus, MRS agar and broth, a Bunsen burner, a striker, an inoculation loop, molasses, glutathione, and petri dishes. Then, before I was able to start my experiment, I had to grow Lactobacillus acidophilus and MRS broth because it was at first a probiotic supplement. I then randomized my sample, which was 32 total, by using a random number generator to select which dishes would get molasses treatment. After randomizing my sample, I swabbed Lactobacillus acidophilus onto the petri dishes, and when swabbing was done, all dishes were placed in the incubator at 37 degrees Celsius for 48 hours. After 48 hours was over, I removed all dishes from the incubator and counted the bacterial colonies found on each one. I recorded this raw data into Google Sheets, which was then uploaded into Data Classroom. I then plotted these points on a scatter plot and conducted statistical analysis. This scatter plot shows the relation between my treatment group, which contained molasses and glutathione, and my control group, which just contained glutathione. As you can see here, the mean for my control group was very low, which means that minimal growth was observed. And on the other side, you can see that there was significant growth observed for my molasses group, which made the mean close to 4. This shows a lot of variance among how many colonies grew in each quadrant. This leads me to conclude that at a 0.05 level of significance, there is sufficient evidence to suggest that the treatment of molasses supplemented with amino acids is significantly different than just amino acids. This means that I rejected my null hypothesis with a p-value of less than 0.01 and supported my alternative hypothesis. This shows that there is strong confidence that molasses helps Lactobacillus acidophilus grow better than just glutathione. This study has the potential to benefit the production of lactic acid bacteria in the dairy industry by lowering costs. In using growth substrates such as molasses, the cost of production is lower because more bacterial colonies are being produced by adding small amounts of an inexpensive supplement. However, I did observe one glaring limitation while completing my methods, which was the lack of growth observed in my control group. Because of this, I was not able to effectively compare the growth between my treatment group and my control group. That is why it is recommended for future research that this study be replicated to see if results can be obtained in the control group. Another outlet for future research would also be to isolate the effects of molasses. This would mean eliminating glutathione from the methods to see if molasses still works to the same capacity. After completing my study, I have learned the importance of the inquiry process and how it allows us to answer questions. It has taught me more about the scientific field and how researchers find gaps and look for answers. Along with this, I have also learned how patient I have to be when undertaking an assignment as rigorous as this one. I feel like I have grown from this experience and feel more prepared to potentially pursue future research in college. Lastly, I would like to say thank you to all the teachers that guided me through this process. Thank you to Ms. Long for giving me all the materials to write my paper and deliver this presentation. Thank you to Ms. Dillon for helping me revise my methods and guiding me through my experiment in the lab. And thank you to Ms. Gravitt for assisting me in understanding my statistical analysis. At this time, I'll answer any questions. Yes, Ms. Martin.
Yes, I was just wondering where you came up with the 48 hours of bacterial growth.
Ms. Martin is asking me where I hours for my bacterial growth. After reading many studies and seeing how long they incubated their bacteria, it was mostly around 1 to 3 days, so I felt that 2 days would be sufficient to see enough bacterial growth.
Okay, do you think that it could have been a change in the growth had you waited 3 days? Just like for your control group, because they didn't have any growth, do you think maybe you could have put them back in the incubator and checked them the 3rd day?
Ms. Martin is asking me if I would have let my bacteria incubate another day, if I would have seen more bacterial growth in my control group. I actually did do this, and no growth was observed after the 3rd day also, so it didn't make a difference, unfortunately.
Okay, thank you.
Yes, Mr. Barber.
For your control variable, you use MRS agar with glutathione. Can you explain kind of what that is again, and why did you use that as your control?
Mr. Barber is asking me what glutathione is and why I chose to use it in my control group. Glutathione is a grouping of 3 amino acids that I read about in a study that helped promote bacterial growth. So I wanted to implement that also into my control group, just to test if molasses would work better.
Gotcha. Thank you.
I have one question for you
Yes.
relating to the non-growth. That would just be an experimental error. Did you find any other or experience any other things that you did not expect with this, or was just the lack of growth from the control?
Mrs. Ashworth is asking me if I experienced any other limitations besides the lack of growth in my control group. That was the only limitation that I